This video shows how to operate BMSeed's microelectrode array stretching, stimulating, and recording equipment, or MEASSuRE, which integrates mechanical stretch, electrophysiology, and imaging. MEASSuRE uses BMSeed's proprietary soft and stretchable microelectrode arrays, or SMEAs. SMEAs remain functional upon an applied stretch, unlike glass MEAs. Before stretching, the SMEA must be sterilized, plasma treated to turn the hydrophobic surface hydrophilic, and coated using standard cell culturing protocols. Cells are then seeded in the SMEA and culturing media is added. The SMEA is then placed in an incubator at a temperature of 37 degrees Celsius and an environment containing 5% carbon dioxide. Once the cells have grown and attached to the SMEA membrane, the SMEA is ready to be stretched. We will now demonstrate how to set up and run each of the 3 modules of MEASSuRE. The mechanics module controls the movement of the cell stretcher using the MOTIONLAB software. BMSeed provides a set of macros, which correspond to different strains, strain rates, and type of stretch, and we are happy to help you program additional macros custom to your research. For example, a cyclic stretch command will simulate physiological cell stretching, whereas an impulse stretch command will simulate a pathological or traumatic cell stretching. Here, the impulse stretch macro is selected. In a separate window, we can monitor up to 4 parameters of your choice during the stretch. Before loading the SMEA onto the MEASSuRE platform, we first lubricate the indenter to minimize friction during the stretch. Then, the SMEA with the cells is removed from the incubator and placed in the recess of the interface board. Next, the imaging module will record the movement of the cells during the stretching using the Motion Studio software. Optimize parameters such as exposure time, gain, and region of interest to produce good image quality during the stretch. The electrophysiology module uses the Intan RHX software for recording and stimulation to obtain a baseline measurement from the cells immediately prior to stretching. We can then remove the excess media to prevent any splashing during the stretch motion. Now that all 3 modules are set up, we can run the macro and several things will now happen simultaneously. First, the actuator will produce the motion of the impulse stretch by pulling the SMEA over the indenter and causing the cells to stretch with the elastomeric membrane. Second, MOTIONLAB records the user's selected parameters, such as position, velocity, and current shown here. Third, Motion Studio detects the motion and begins recording a video at high frame rates of the SMEA and cells during the stretch. The macro execution is now complete. Replace the culture media in the SMEA and perform post-stretch electrophysiological measurements with Intan RHX. The SMEA can be stretched again, swapped with a new SMEA, or simply returned to the incubator for later use. Finally, the user can examine and save the following data from each software: electrophysiology data from Intan RHX to compare pre- and post-stretch cell health, position and velocity data from MOTIONLAB to determine strain and strain rate, and the recorded video from Motion Studio to verify the actual strain and strain rate that the cells experienced. All 3 of BMSeed's MEASSuRE models are operated similarly, but have different specifications to target different applications. MEASSuRE Premium and MEASSuRE X can produce high strains at high strain rates, while MEASSuRE Mini is best suited for cyclic stretching in an incubator. To learn more about product details or potential collaborations, contact us today.